And we have the bottom in sight, actually.
So while we're getting set up on the bottom, we had a couple other people join us on the line. Would you guys like to introduce yourselves?
Yes, this is Tom Harrigan from NOAA's Deep Sea Coral Research and Technology Program.
Good morning, Tom.
Morning.
Glad to have you with us.
Oh, I also had a note on the small unbranched primnoids that were observed yesterday and on the deep dive. I don't believe any were collected.
That's right. We thought that we might have had one attached to the last collection that we scooped, or maybe it wasn't the last collection, but the one that we scooped, and it turned out we didn't collect it. So that was unfortunate. We looked all over the place. We sieved the water to make sure we didn't miss it, but we weren't able to collect it. If we see any today, we will definitely collect it.
I think that would be good. In the discussion, it was initially noted as perhaps Paranorella, a genus that was identified from New England seamounts. That Paranorella is actually a branched species.
Oh, okay.
This is more likely to be Convexella, which was also identified from the New England seamounts. And the species that was identified there also occurs on the mid-Atlantic ridge, but it had not been observed below about 2,600 meters.
Oh, that would be a depth range extension then, huh?
Significantly deeper, also significantly further south. And that genus is also the genus which has the deepest primnoids, and I think the deepest gorgonians, alcinaceans of any kind, down to abyssal depths.
Well, since we're in a similar depth range today, keep fingers crossed that we'll come across another one, and we can collect it to verify all of that.
Okay.
Tom, which coral did you say this was? What was the name of it?
It was Convexella.
I haven't even heard that genus name before.
What? Sorry, I didn't hear you.
Oh, I haven't even heard that genus name before, but then again, I haven't worked in depths like this very much ever, so.
Yeah, they're unbranched and look very similar to what we saw yesterday.
Yeah, I think that's a good point, Tom. I'm looking at the image of the Convexella that we collected on Yakutak Seamount, which is just north of this area of the New England Seamounts. And if we see another one, it should be recognizable by the pattern of how the polyps arise from that branch. So that's a really good suggestion. That one that we collected was from 2,400 meters, so you're right, this would be significantly deeper. The Paranorella had come from 3,500 meters, which is why we did suggest that one yesterday. But you're right, that one branches, and it branches from pretty close to the base, so you'd expect one of the size we saw would have already been branching.
Yeah.
Okay. We were able to collect that bryozoan. We did have that in the collections on the ROV yesterday, so that was exciting, and we got some good images of it, but it was so much smaller than I thought it would be. Again, these images that we see the video, I always have to remind myself of the size of the scale with those lasers, because things they look bigger on the big screen.
Earlier you asked about possible collection targets for today, so we're in an environment that we could find Okeanos arbuscula.
Okay.
Be a good connectivity target.
Great.
And Tom, the Convexella, is that also a geographic extension? Has it been noted this far south?
Yes, it would be a geographic extension.
Thank you.
It has not been reported from the U.S. southeast or any place southeast of the North Atlantic, further north.
Well, thanks, Tom. That's really exciting.
Yeah, it is. And I'm really sorry that that one that we tried to collect yesterday didn't wind up in our collections.
If you are just joining us, we have only very recently landed on the seafloor. This is our third dive of the mission, and we are at a depth of, where am I looking?
3,345 meters.
Yeah, I couldn't see the monitor. So this is a comparable depth to the one of our dive yesterday. We're in a slightly different type of setting. We are farther south. We're on the Blake Ridge off the continental margin of the southeast U.S. coast. This particular area is an area of very large undulating bedforms. And we were sort of at a loss as to what to call them because they are so large. Think of large sand dunes on land. They're formed by wind blowing. These are gigantic compared to those, any sand dune you've ever seen on land. These have distances between the tops of the dunes between 800 and even 1,000 meters. So they're absolutely enormous. They were not formed by wind blowing. They were formed by very strong currents over long periods of time that have shifted all these particles of sediment that are on the seafloor. We haven't gotten a close look at the sediment yet. The ROV is still getting oriented for the day, and we haven't actually begun the dive track. So I'm sure we'll have plenty of opportunity to get a zoom in on these sands to see if they are in fact sand or mud or made of microfossils. Of course, if they're microfossils, we won't be able to see that on the video, but we'll get a collection at some point.
Yeah, we, with that scoop sample that we did yesterday, actually provided a nice sediment sample as well.
Yes, I was very happy.
The mud was so sticky, no wonder why they couldn't get it off the scoop.
Yes,
It was
yeah.
kind of crazy.
What did I call it? It was the consistency of peanut butter and the color of coffee ice cream.
Yeah.
Yep, so that'll give you a nice visual. And I said that, and Cheryl said, "You must be hungry." It was, in fact, right before dinner. And this looks very similar, but we won't know until we actually get a feel for it. Looks like there's something in the base of that.
Go ahead, pilot. Roger that. What do you think? Leslie, would you like a sample?
I think we should at least take a quick view.
Okay, quick view. Roger that.
So we've seen features like this, particularly on the first dive at a little shallower depths. And one of our call-in participants or chat room participants suggested it was from beaked whales foraging on the seafloor, which is pretty impressive. We get a nice little cross section. That's quite a sharp. That doesn't look whale made.
No,
It's a little sharp. It looks more like a grab sampler made that, or some kind of apparently we did not make that, but oh, we can see some little chips. That might be a good little sample to get because there is some consolidation of the sediment here. So if we had a sample here, we probably wouldn't need one later on.
Sounds good.
All right. Maybe pilot, if let me communicate with the pilot that.
Yes, please. Would it be possible to use the scoop and get some of the sediment? So while they're setting up, it looks like there's some sargassum at the bottom of the monitor of the screen right now, and we have been seeing sargassum at the surface in this whole area that we've been in, and that's basically over 3 kilometers above us. So this is a way or an indication that surface productivity reaches the sea floor on a fairly regular basis. We saw that patches of sargassum all day yesterday during our dive at a similar depth, and as you can see, there's not much obvious food down here, and so a big patch of sargassum like that is probably quite a buffet for somebody coming along.
The 2 red dots are the laser beams that we use as a measuring device. They're 10 centimeters across. We don't keep them on throughout the dive, but it's handy to get a sense of scale, particularly of the organisms, because it's very hard every time we bring up a sediment sample, or actually every time we bring up a biologic sample, we are amazed at how small it is. With this incredible zoom capability, we often think that things are much larger. So the manipulator arm is grabbing our little scoop device, which served us quite well yesterday. I'm not getting.
Shoreside, you may also take note whenever we do samples on the stream 3, we change the quad split, so you've got 4 different angles of the sample process taking place.
As I mentioned just a few minutes ago, with these large bedforms, this site, by the way, was proposed by Roger Flood of Stony Brook University, and we're hoping he'll get on the line at some point to give us a little more information. But these very large bedforms don't occur overnight for certain, so it tells us of a long history of fairly high and consistent velocities of currents, of these deep water currents. So we are about to jab that scoop into the vertical face there. We also heard from Scott France earlier that, and maybe you said this, Cheryl, when I wasn't listening closely, but Scott France mentioned that he didn't think beaked whales got this deep. And again, it does not look like a beaked whale. Pilot, if you can go as deep as possible, that'd be great. That'll work. And this will be a very nice comparison to yesterday's sample, which was farther north. Yesterday we experienced some fairly swift currents, and they were coming from the north as expected. They're part of the western boundary undercurrent that flows southward. It's very cold waters. This sediment's nice. It has a good consistency to it. All right. Thanks, pilot. I'll do. If you would. Yesterday's sample that looked very similar, but who knows if it is similar. Yesterday's sample was washed, and all the clay-sized particles, the very fine particles that when you put them between your fingers, it's silky. You don't feel any grit. That's clay-sized particles. And so we have a sieve on board that we can wash that mud out of the sample. And it takes a long time, especially with this cohesive mud. And eventually leaving behind anything that's larger than clay size. And we had, oh, excuse me. If we can shake it, that would be great. Yeah. So the sample just, most of it fell out of the scoop, which is good. That's what we want. Because we might need this scoop later to scoop up some organism that is on one of the lonely rocks out here. And that's going into the port outer bio box. Okay. I'm sure we got plenty which we can use if you can just give it another tap or shake. I saw a large chunk fall through. You don't need too much of this to be able to characterize it. I'm not, I don't have the screen up. Oh, yeah. Yeah, that would have been nice to get that because it's a little older sediment, but I don't want to forfeit our scoop for the day. Okay. Is there something you can tap it against? Not the bio box probably, but to loosen it? Oh, you can do that? Okay. Okay. Pilots trying to shake off the sediment from the inside of the scoop. It's cohesive enough to still stick to it. I read somewhere somebody characterized Blake Plateau sediments as being the consistency of toothpaste. Oh, yes, he got it. Valiant effort. Excellent. So thank you. Okay. Great. So I'm happy. I believe there was also sorry. So nearby there was a piece of sargassum upper right of the screen that we might want to take a peek at. It seemed to be substrate for an organism. The lack of rocks available, they will probably cling to anything. So while we're packing up, I'll just finish up with the results of the sediment sample from yesterday, where it was about 80%, at least 80%, the fine materials that were sieved out and washed away. We did preserve some raw sample for those of you interested, of course. The remaining sediment on the screen was very, very fine still. It would feel like salt or sugar size particles to the feel. We call that a silt size, well, even smaller than that, and it was a silt size particle probably. And we have a microscope on board, and we'll take some pictures once it dries, but it was almost completely comprised of single-celled organisms that secrete a calcareous or calcium carbonate shell, and they are known as foraminifera. We have a great visiting rat tail. Do we have an ID on that, Cheryl? I'm sure we did from yesterday.
Coryphaenoides rat tail is what we saw yesterday, and this looks very, very similar. So although I'm not a fish biologist, if there's anybody who has a different ID on it, please let us know.
We're going to take a peek at this organism, upper right, out of the pit. We probably should take a look at what's in the pit too before we leave. We didn't do that, but certainly wanted to take a look at this.
And it does look like a sponge. Wrapped in sargassum. So there must be a little
there must be
rock
a rock under there that the sargassum is attached to?
Yeah, that's what I was thinking.
I can't see how a sponge that large could just grow on sargassum.
Maybe it's an old tube of a polychaete or something. that it's
that's
growing
the
on.
ultimate, or maybe that's the sponge stalk. I'll bet this is a stalk sponge, kind of like a hyalinema. And we're seeing
Yeah, there's a
the
stalk and
we get
a
close-up on that.
branch at
So
the
Oh.
top of
wrapped
it as
around
though
silica
it's growing.
spicules.
And there's a stalked organism to the lower left of the screen.
Oh yeah, that's one of the polychaetes,
Oh, okay.
I think.
Oh, they're living in the sand.
Yeah,
On the substrate.
they have a tube. We saw some yesterday. They have a long tube, sibellid polychaetes, I think that was.
You can see at the top of this sponge in the center, it comes to a point,
Yeah.
and that's the up part of that stalk. The stalk itself is a bunch of very long glass silica spicules that kind of are wrapped like a cord to form that stalk, and then it goes into the sediment and they splay out to anchor it.
Is there interest in a sample, or should we move on? Or
I'm not inclined to call for a sample at this point.
yeah, it seems early.
Yeah.
I'd hate for this to be the only one we saw of this species, but we're not trying to collect one of each species either.
So while we're repositioning, Scott France's wife, Patty Russell from NOAA Fisheries, commented on the beaked whales. And most whale feeding is above 1,000 meters depth, but she did have this for sperm whales. Maximum dive depth attributed to sperm whales include 1,135 meters, 1,827 meters, 2,250 meters, and 3,195 meters. But note that those are extremes, and sonar tracking has shown that 99.9% of dives are less than 1,000 meters.
So what we're seeing is apparently sargassum that has sunk to the sea floor. This normally is a surface floating form of seaweed. And it is a seaweed, correct? I'm not saying the wrong thing. And it has those little flotation nodules on it. I'm sure they have a name.
Oh, yes, they do. I forget it.
Okay,
right
sorry.
now.
I think we're good here. Let's begin the actual transect unless there's objection.
Channel turned on their video or on their computer, then I won't come through. If they only have your channel
We
turned
see
on.
some organism here.
So I actually had that turned off during the science meeting and forgot to turn it back on, so the pilots are now back on the right
That was
channel.
an error on our side. Ambalula, maybe.
This is possibly our first sea pen, an umbellula.
Definitely telling us the direction of the current.
All right, video, you can start in. I get it.
It's beautiful seeing the internal organs of this organism. So can you tell us a little bit about this? What
Are you ready for a move?
group of organism, what phylum it belongs to?
Go Pat is. Up to Pat.
Sure, so we're looking
Bridge,
at
are we now?
Sorry, video, he's just
well, the genus name
wiggling
is
in the wind a
umbellula,
little bit.
And this is a type
Yes,
of
can we repeat that last move,
octocorals,
please?
And it's within the subgroup of octocorals called the Penatulaceae, which are the sea pens.
Good copy,
And
thank you,
umbellula
Bridge.
is pretty common. I'm not surprised at all to see this. It's very common at very great depths in soft sediment. You can see it's got, what, 1, 2, 3, 4, 5
the
large
lasers.
polyps there. They can have
Lasers,
more or fewer polyps than that. But you
Let's
notice all the polyps are concentrated
fog light
at
action.
one end, and otherwise has this very,
Far
very
right.
long stalk. It's called the radius.
This kind of
It
in
has
this
a
kind of
single
scenario, it lights up the
Bridge.
sediment a lot
hard
too.
skeleton in there.
That same.
And then at the bottom it has
Oh, the shadows are
anchored
kind
in
of fun.
mud by
I don't know.
inflating
Up
the
to
soft
video.
tissue at the
I think
bottom.
you actually need to be about another foot and a half, 2 feet closer before that light would make that difference.
It's it's a
okay.
group that's
Copy.
still got a lot of
If
questions.
you turn it
with
on, you can see it in the
One thing we know
ROV
is many of the
Yeah, it does.
species that
It's
are
pretty
called
hot right
umbellula are
near
not
us.
in fact umbellula. They belong to other groups. There's still
Well,
some
then I'm
work
all set
going
when you
on.
are.
Video's
Within the
clear.
last couple of
Alright, off
years,
we go.
there's a new genus called
Somebody
Porcupanella
floating
that
through.
was identified. And right now, I wouldn't know the difference between seeing them in video if it was one versus the other.
Thanks, Scott. For our listening viewers, that's Scott France. He has been on several of these expeditions.
Fish
and
coming
is
up.
one of the, well, is
Confirmed.
the onshore science lead coordinator. I'm not sure of your official title, Scott. Can you tell us, please?
Can you get a snap and
Yep.
catching for a
Sure, let's
potential
see.
highlight?
I'm serving as the science advisor for the Okeanos Explorers
Science
Atlantic
advisor.
Expedition.
Right. And a wealth of knowledge. Thanks for all your input.
Try not to laser me in the eyeballs.
And to your wife for the beaked whale information.
There
Thank you.
go.
She is
Lasers
still
on.
researching that, and she's now reading about some from the South Atlantic that have been known to get to, believe it or not, 5,000 meters. So we're following
Well,
up to
if it's
see
Interesting.
exactly
Yeah, go ahead.
what the deal is there, what sort of evidence was used.
Oh, he's going right into my dust cloud. And I'm making more dust. Oh, my laser bias set a little high.
This is another one of the Coryphaenoides rat tails. They seem to be the most common fish that we've seen at this depth, both yesterday and today.
Well, they must be feeding on something, so maybe we'll see
Black thing just off right.
some of their food source. Even though the
on the
landscape
bottom?
here looks
Yeah.
extremely flat, which of course it is, it's part of a very large bed form that you can't even see at this scale. It's so large. We're climbing down the edge of a smaller of two sand wave, mega ripple,
Yeah, you can come
giant
in.
dunes.
See what it is.
We're not even sure
Fish
what
or
to call them.
garbage or what.
But they're very large features, so we probably
like a
won't
piece
even
of seaweed.
notice that we're going up and down.
It looks like a piece of seagrass there.
More of those gouges.
And these pits that you see here and there
But you're starting to.
with the big sort of levees of sand around them, we're looking into seeing if they might be depressions made from beaked whales feeding on the seafloor. We've seen
Probably
them in
also, we need a
shallower
little
depths, and
Port, I can rotate if you want,
Leslie, can we get the lasers in this pit so we can get an aside?
Sure,
idea of the
sure.
size of it?
depending if you want to investigate these trenches.
I'll take a look at this one and I'll get back on track.
Did this twice now. Sirius does.
Yeah, it's a glitch on Sirius.
Yeah.
Known error.
Copy.
Not sure if it's the
It's another sharp-edged feature. It's really
thrusters
interesting.
themselves are erroneous.
Pilot, could we swing
So
around
one of
and
the things
look at
that's
the
different
face
about
of that
this pit from the ones that we
ridge,
were
maybe?
seeing, a
Sure.
couple of days ago, is that it isn't clearly being started at one end and pushed with the furrow ending up at the other end.
Yeah. I was noticing that
Looks a
also.
little bit more like something's flailing around in the bottom.
So this
And
puts the
it's
current on our back, so
Bigger
any mud
than
on me
usual
is getting blown in
burrows
the
without
screen.
any kind of canal
we can
going
do
into
this
the
to take a
sand,
look
so
for a
it doesn't
second.
look like a burrow of any type.
The squareness of it is very surprising. It's
Yeah,
if we
very
weren't
angular.
at 3,300 meters, I would think it was from a sediment grab sample, which
Yeah,
is a very squared instrument that just from a ship is deployed, and we take samples.
Yeah, box core.
Yeah, I just can't
Yeah,
imagine.
I was going to say something similar but crazier, and that is how long would depth sounding marks last in this
I
sort of
know
environment?
it's.
Other than
And how likely would it be that we happen to come across them?
heavy weight here, but that was an idea.
And
Yeah.
give a snap on the white thing just beyond the hole.
Yeah, I did see that.
Crazy.
We can go for it if you want a quick snap on it.
Well, if we keep seeing them throughout the dive, they're not box scores.
There's some organism
Oh,
back
yeah.
here. Ooh, a glass
Glass
sponge.
sponge.
Yeah. Hexactinellid glass sponge. Vase-shaped. Sometimes these have a pair of shrimp that live inside. But there's something in the middle, but doesn't look like shrimp from here. And that's a very small one.
Sorry, it's kind of weird
And then
flying
there's
with your
something
butt to the
behind
current.
it that has 3 branches. Do you see that, Scott?
I am seeing it now. Really small.
Yes, very small.
And I'll tell you, my first instinct on seeing something that's that small and kind of fine branches is a carnivorous sponge.
Try and roll,
laterite
sorry.
Sponge.
Oh.
So that also supports that ID.
Video's clear.
Okay. Pilot's clear.
Yeah, in the
Bridge.
chat room, Tina said that the
Move
small
complete.
branched,
Copy that,
3-branched
Nav.
organism we were looking at is likely a bryozoan. But then others are saying it could be a sponge. I think we've had this conversation a few times there. We can't figure it out even with a whole bunch of experts.
Guess next time we see
Yeah,
one
one of the interesting
those, if we
things
could
about
just sort of
those
sit right on top
observations
of it.
is how much convergence in morphology there is
That's
among
a
different
good set of
organisms.
view.
Yeah.
And that's because they're all
Coming
surviving
around.
in a similar habitat and have the same problems. They have to get food. They have to get food out of the water column. And there are some very efficient ways to do that. And so different kinds of animals may converge on a similar body shape in order to survive here.
Yeah, great point, Scott. This is not an easy place to live. And Tina commented that carnivorous sponges like harder geology, bigger rocks instead of, but this is probably just a little tiny pebble or something that it might have grown on. Or I don't know if the hexactinellid sponges, if they need any hard substrate. Would you know that, Scott? I would think so, but. And then if the carnivorous sponges can just settle on the sedimented surface. They don't need hard substrate.
Sorry, I was typing in the chat log there. So right, that's a good point. But my response there was, well, wouldn't bryozoans require the same thing? A lot of these
I just don't want to hog it.
animals, these colonial animals, well, the sponges aren't colonial, but they grow as some of them grow in these branching forms that look like a colony. But they start life as a larva, and the larva can be swimming.
Go ahead, watch it. You're a little quiet, but I can hear you.
Typically, they do require something hard on which to settle and begin to grow and metamorphose. You can picture a caterpillar that's turning into a butterfly. The caterpillar finds something hard to attach its chrysalis to, and then it transforms into a butterfly. These larvae will find some small
I have to
hard substrate attached to
be
it.
preemptive about slowing down in this kind of condition, so I try to slow down and then wait to hear if I see if I hear anything, and then if I don't, move on.
Try that.
But now I know I'll just take zooms.
Yep.
And if you could try to get them between the skids.
With all the mud rolling, I don't know if you're going to really want that, but we can try it.
Yeah, you want to come up and give me a break real quick?
Down into the mud as an anchor, so it could be that way for the cladorized sponge.
I'm facing into the current now, so it should go a little better.
Guessing based on limited information from our collection.
Right. Reminds us how much we still need to learn.
I'm going to get another move in.
Especially at depths like this.
Yeah. I'm good with one.
Fred,
and you're at 3,340 meters.
Repeat the move, please.
The ROV is currently facing directly into the current, which is coming from almost
Good
due
copy.
north, flowing southward. Take a zoom
All
in
right,
on this
Biddy,
baby.
come in.
Oh, that's another one of the
Another one of them.
hexactinellids.
Copy, move initiated.
They are so beautiful.
It's a nice different view from the previous one. You can move right down into it.
Is that an amphipod or two on top? Oh. What is that?
Something fuzzy on the inside, huh? Or spiky? Hold on. Oh, sorry, video. This current is unforgiving. Come out a little bit.
Certainly expect to see
Let me just put my toes right down.
I can't quite make it out on my screen. I'm dealing with a slightly smaller screen today than yesterday.
All right,
Yeah, it almost looked like more of an isopod than an amphipod when we got close. But and when there's a hermit crab.
Oh, the contour lines are on.
Yes. It's moving.
It's quite flat.
Well, that's a great view.
Look at that inside.
What is moving inside there?
That? Is it a scale worm that might be migrating around? Yeah, it looks like it's mobile.
Want to try the extra lights?
Sure.
It does
It's pretty
look
bright,
like
it seems.
I think it
Yeah,
is.
this case is a different.
Fog lights
Okay.
coming on.
Looks like they have lots of fur on the dorsal side. The elytra are modified into these dense chetae that make it look like fur. I wonder if a larva got stuck in there and grew.
Huh. And then there's the amphipod walking around the edge.
There are isopods that are on the sponge.
Okay. Yeah, so the scale worm
Wanna
is
shoot it with the still a
inside. You
couple
can see it
times?
moving,
How do you
coming
do
out
that?
to meet us,
Thank
Looks
maybe.
like the tongue.
DSC.
Or is it
There's
on the
one DSC.
underside? I
Yeah,
guess it's on the
just-
underside.
Huh?
That's
Press
on
it.
the
Just
inside.
leave it
Oh,
on
it
for
is
a
the
little
No,
while.
I think it's the
Copy.
inside because that's its back.
Okay.
I
Wow,
mean,
that's amazing.
Looks like it.
So
That
for the
is very
still,
You're seeing the crab
is that
below.
about where you want it?
Yeah,
then?
it's
Yeah,
the
let's
hermit crab.
zoom in on the
tip of the
crab.
toes in
Maybe
between.
see.
Should be an input there,
It's
yeah.
got
Oh,
a
it looks
nice
like
little
he-
gastropod
Yeah, you can-
shell.
There you go.
Yeah, it looks like it
yeah,
has
it's
some
way
air
out of frame.
conditioning, though.
Windows to
Too
the deep.
flat.
Yeah, a
Yeah.
little broken up.
All right, you can kill it. Oh, you going for the crab, Roland? Sorry.
We were getting both.
Do you want your Zeus back?
Video's clear.
Okay, pilot's clear.
Let's go full screen once. These lights.
You can see almost a root system But that's just old sargassum, probably. Is that what you think the base of the sponge is?
You know, the two brown spots on either side would be sargassum, but I think that those structures are, yeah, the base of the sponge. Scott was talking about the extended spicules.
So that those are serve as
the
sort
anchor.
of roots into the substrate, yeah.
Okay, video's clear.
Pilot's clear.
So even one organism like this creates a little oasis for other organisms to get off the sea floor.
Perfect placement of that light there.
You want to kill the light for now?
Light coming off.
We're seeing some interesting trails, too, that we haven't seen on the right of the screen. You see that black, meandering little line. Some organism has left its mark on the
Bobby's going to take it. Give us a second.
fine-grained substrate. So we're looking at a vast field of mud, basically. And in marine geology, we actually use the term mud to characterize the seafloor. Mud is a combination of silt and clay-sized particles. Clay being the very finest, like what you think of with modeling clay, and then silt being just slightly larger so that you could actually feel the grit between your fingers. So mud is a combination of those two grain sizes. And that's what we see here. Most likely. We'll find out for sure after we get our sample this evening.
It was so interesting to see the sieved forams yesterday. That they were just so perfectly round and lots of different colors. It was amazing.
Watch lead, pilot?
Go ahead, pilot.
Yeah, we just did a switch up here in the front row. I heard there was something on the bottom you wanted to look at.
I think we were ready to move forward.
Roger that. Moving forward.
Talk about the black trails.
Yeah, we might want to zoom in on a black trail that if we see another one near a site that has some organism to take a peek at.
Roger that.
Especially if we see a black trail that ends, perhaps we can see who made the trail.
So one difference from where we were yesterday, we were seeing a lot of the heart urchin tracks in the sediment, and I haven't seen those so far, or they're not as numerous anyway.
Quick zoom here video.
Oh, we have a barnacles.
Oh yeah, gooseneck barnacles.
Aren't they beautiful with their appendages? Those are their feeding appendages?
Yes.
That's a beautiful shot, video. Thanks. Would these be considered gooseneck barnacles?
Yes. So you can see they have a long stalk. And there's a species of these that hitchhike on whales all over the world. And I guess
like they've really bowed that sponge over. They must be growing on the tip of the stalk of that sponge.
And this would be another stalked sponge, right? And they're growing off the stalk.
We saw earlier that said the hyalinema, that you could see the point at the top of it. So I'm assuming the barnacles started settling on that upper point, maybe, and stripped away some of the sponge tissue. That's a guess.
Huh.
Oh, actually, I have it backwards. So they're on the base of the stalk, and the sponge is sticking towards us, and that pointy tip has become exposed. You can see the frayed apart sponge spicules that make up that stalk.
Yeah, so would that be predation, that somebody ate part of the sponge, and that's why it doesn't look complete on top?
Yeah, it's a good question. I don't know. Another possibility is that maybe as those barnacles are getting on it, it's weighing it down, and so now it may be hitting the sediments, and so it's just being abraded with the sediments.
Back to this point.
Oh yeah, that could be true.
All right, pilot.
Pilot's
just
clear.
About looking down at you.
Video's clear.
All right, let's come on.
Martina Moldovtsova noticed that there were
Bedfellow
hydroids of the
mounds.
barnacles
It's
as
in a
well.
line.
It certainly appears that some organism is making these pits, and other organisms appear to be taking advantage of that. Let's see deposits of the sargassum.
Yeah, they seem to
They roll
wind up
into
there. Tumbleweed.
Yep.
You guys ready for another?
That one looks a little bit more like the
I'm looking
beaked
at
whale
pilot
scar in
about
some ways, where it was
5
pushed.
degrees. So I think we could do another one.
I agree.
It's pretty
Definitely
flat.
looks like something got pushed into that.
Mm-hmm.
Bridge, ROV nav.
If you want to lose the skids, you can video.
Another ship move, please. We'll do an additional 30 meters at 350 degrees, speed decimal 2. That's affirmative. Thank you, Bridge.
Almost looks like footprints.
Thank you, Bridge.
It's another sponge-like organism.
Go ahead, video
Maybe
starts
that
going.
isn't sargassum. Oh, yes.
That's a different sponge,
It
though.
is a different sponge.
I think there's going to be
That's
some interest in
interesting.
the sponge
I
down
don't know
below
for
it as
certain. I'm
well.
not a sponge expert, but this looks similar to the one that was on the stock, except it's all the way down at the substrate.
Yeah.
I agree with you that it's not the same, but it has that impression. So I wonder. I think we collected something like this last year, asking the same question if the stock was just more buried, but that wasn't the case. It looked like a very small sponge there. Yeah, that's a newly growing one, maybe.
Oh, that's so amazing. Look how tiny that is, this little bitty stalked sponge on the lower right.
Yeah, they got to start off small at some point.
Yeah.
Kick on
Pilot's
the
clear.
lights.
Yeah, so
What'd
closer
you
look
say,
at this.
So you
The
wanna
other
kick
one
on the lights?
left,
No,
that's
let's move
definitely
on.
different from the others.
Yeah.
Be another sort of maybe a demo sponge. This one, I'm not sure.
Maybe a little tilt up. There we go.
And more sargassum. You can see that we're gently climbing now up one of these sand waves.
Copy that. Thank you, video. Does feel good here.
Just do a quick snap over here, video.
Pink thing.
Yes. Hermit.
We're at 3,342 meters. Ooh, and there's a hermit crab. Now, hermit crabs are decapod crustaceans, which means deca, they have 10 legs. But hermit crabs have their fifth pair of appendages highly modified. Then they're tiny and they hold on to a shell. And so the inside of that hermit crab is a very soft body that curls to match the curling of the gastropod shell that it lives
Sorry,
in.
video.
And there are some
That
hermit
last little
crabs
bump.
that
You can go back
have
in.
a straight abdomen and live in things like polychaete tubes and, yeah, especially polychaete tubes that are kind of look like straws. but they're, of course, biogenic, made by polychaete worms.
Is that full?
No.
And we've seen
Was that counting
several
for a
cases of sea anemones that live on the
Just need this rotator to
shell
work
of the
better
hermit
for me.
crab. So that's quite an advantage to both species, because the sea anemone has stinging cells and it could protect the hermit crab, and then the hermit crab gives the anemone a lot of
All right, let's
mobility.
come wide.
Cheryl, I'm thinking this one might be carrying a zoanthid, and I'm saying that because it looks like pointing forward there was one little nubbin, looked like a round hole, and then behind it I could see some tentacles, and that would suggest there are separate polyps that open up.
Oh.
Also a relatively common association with epizoanthids.
Good eye, Scott.
Let's go partial. Let's see. Pirouette.
Sargassum on a sponge.
Yep.
Stock.
Another stock sponge. Okay. Come wide.
Seems that wherever there's an organism
up
or an
there.
indentation, we'll find a piece of
Sorry?
sargassum
You
that
see a ledge up there, maybe a mound.
Yeah.
He's not showing up on your sonar.
So is this getting to the top of one of these waves?
Yeah, it's looking like we might. I'm not seeing the track.
Swimming
we are.
something.
Yeah, we'll just let him go through. Scale worm.
Okay.
So this ledge is on the scale that we won't see it, I don't think, on our mapping. But we'll see what's on the other side.
Kind of
Kinda
looks
see
like
it
the
in
surface of the
Sirius.
moon.
Very much so. That's great. Now you get a sense of there is undulation to this landscape, or seascape. And how large these bed forms are. These are enormous dune-like features that were formed from flowing water as opposed to windblown.
What's the strike on it?
62? 60, yeah.
You just are
Nice
teetering
view of
off
the
to
crest
my
of
port.
one of these giant sand waves, or mega ripples.
And it sure is impressive when you look at the multi-beam map of this area to see how extensive these are.
So we're following along the ridge of a small one. So, and these are probably features that are smaller marks, smaller bed forms on the larger scale features. Well, I'm sure they are actually. We wouldn't be able to see the crest of the so isolated like that. You get a sense of the current here. Any of those little gravelly pieces of sediment have a trail behind them. We're going right into the current right now. So you can see the trails left
It's
behind.
like an umbellula.
Confirmed.
Just grab a snap just for science.
Oh, another umbellula. Type of sea pen. This is the second one we've seen this
You guys
dive.
ready for another?
This looks very similar to the other one that
Pilot's
we
clear.
saw.
Co-pilot.
What was the time between the two?
Between
The last call?
last call was 15:07. It ended about a minute ago.
Seemed about a minute ago.
It's a fairly strong current, so
fine.
depending on the position of
Is
Sirius,
it a man-made object?
The last one we
Other-
let settle.